Recording in progress. Buenas tardes. Bienvenidos de regreso después de la semana de descanso. Realmente es un placer para mí presentarles al doctor Simon Alberti. Él es biólogo de la Universidad de Bochum, en Alemania, y su doctorado también lo hizo en biología en la Universidad de Bonn, y permaneció en la Universidad de Bonn un año como postdoctorado, para después ir al MIT, al Whitehead, a hacer su primer postdoctorado con la doctora Susan Litwit. Y estuvo en Estados Unidos desde 2005 a 2010. En el 10 regresa a Alemania al Max Planck Institute en Dresden como joven investigador independiente. 5 años más tarde se promueve a ya un investigador senior, y en el 2000, en el 2022, 6 años después, se convierte en el director del Centro de Tecnología de la Universidad Técnica de Dresden, donde tiene esa posición desde entonces como ya full profesor. Y bueno, el interés de Simon ha sido entender cómo la célula responde a diferentes condiciones de estrés, y cómo a través de estudiar estos fenómenos encontró que la formación de compartimentos sin membrana juega un papel importante en estas respuestas a través de la formación, de la separación de fases. Y es la que he estudiado durante muchísimo tiempo, y ha sido un investigador muy productivo. Ha publicado alrededor de 95 artículos con más de 28,000 citas, con un índice H bastante alto. Y de hecho esto le valió ser el investigador más citado en el 2021 y 2022. Y realmente su trabajo lo pueden consultar, está publicado en las mejores revistas científicas. Y bueno, hoy el título de su presentación tiene que ver con condensados biomoleculares y su papel en la respuesta a estrés y en la enfermedad, como vamos a ver más adelante. Thank you very much for accepting the invitation, and please.
The introduction, I've never been in Spanish, so this is the first time for me. I didn't really understand much, but I could guess somehow what you said. Yeah, thank you, and it's great to be here. And yes, so I'm going to tell you about this new, really 10-year-old idea of biomolecular condensates and what role they play in biology. And I guess many of you have already heard what biomolecular condensates are, but I'm also going to introduce this topic. So first of all, when you think about cells, I mean, cells, of course, have many, many compartments and organelles. And the compartments that you're all familiar with are these compartments that have membranes around them. Yeah. But there are actually a lot of compartments that do not have membranes around them, and they're called membraneless compartments. And you see a bunch of examples here. So these are, this is an immune cell, a T cell, and you have these microclusters of T cell receptor on the surface. So this would be one of these membraneless compartments. You also have centrosomes, which of course nucleate microtubules. You have nucleoli, which are known to put together ribosomes, so they're assembling ribosomes. Again, these are membranous organelles. Then we have P-bodies or processing bodies. These are RNA and protein containing granules, which are thought to be involved in the degradation of RNAs. And then we have in oocytes actually here these structures which store molecules for longer time. And you also have stress granules actually. So I'm going to talk more about these stress granules today. So these are structures that are forming upon stress, and they're also RNP granules. So they contain RNAs and RNA binding proteins mostly. And they are thought to be involved in translation regulation. Yeah. So these are just a few examples to give you an idea what this is all about. Now, and for a very long time, we've known that these structures exist. Yeah, we've seen them by microscopy, but we didn't really know how these structures form. And if you want to understand how these structures form, you have to actually turn to physics. Yeah. So, and of course, you're all familiar with the phase transitions of water. So water can be in 3 states. It can be in this gas-like state here, in a liquid-like state, or a solid-like state. And of course, these are macroscopically really big differences between these three different states. Yeah? But actually, what is underlying this is just little differences in the interactions of water molecules. Yeah, so they are very ordered and strongly interacting in a crystal-like fashion here in the solid. You have more weak and disordered interactions of water molecules, which then give you liquid, and there's very weak interactions in the gas-like state. Yeah, and of course, you all know this. And I think what is really new now here, what we've learned in the last 15 years or so, is that proteins or other biological macromolecules can also undergo phase transitions. Okay, so you can just take an idealized protein, which is here in a mixed, it is well mixed in a solution, and then there could be some kind of change in conditions, such as a change in pH or a change in salt, for example, or a post-translational modification. And then somehow these proteins, they have a higher affinity maybe for each other. And then they start to assemble into these higher order structures, such as filaments here, also these highly ordered crystals, then gel-like structures. There's also glassy structures. And there's, this is the most important one, actually, these liquids. Yeah, so this is actually what we call phase separation. So where you have a separation, well, initially you have this one phase system, but now you have a two-phase system. So you have a dense phase and a dilute phase, which are still in dynamic exchange, right? This is similar to, let's say, a salad dressing, vinaigrette, for example, you know, oil and acid, they wouldn't mix actually. And so that's actually the same phenomenon that we have here. And so, I mean, that proteins can do this has actually been known for quite some time. I mean, if you work in the food industry or so, of course, people have known this. But I think what is not new here is that we more and more, yeah, really see that proteins have sequences that are tweaked by evolution so that they can actually undergo these phase transitions. Really, evolution is acting on protein sequences and designing them so that proteins can do this. And this, of course, we also think is important for function, but then also, of course, for disease, as I will show you later in my talk. Now, when we got into this field, we were initially interested in these ribonucleoprotein granules, or RNP granules, two of which I just already mentioned, these P bodies here and these stress granules, and there's also these neuronal granules in neurons. Yeah, so these are transport granules that are important also for local translation. And so these are very dynamic structures. So actually, when you photobleach, let's say, protein, they go in and out very quickly. So there's actually thousands and thousands of molecules of RNA and protein, but there's no membrane here, right? So this is the really unusual thing. And really, we didn't know for a long time how these structures form. And so then we got interested in this phenomenon, and we actually focused on one protein that resides in these RNP granules, which is this protein FUS, or fused in sarcoma. And it's a very interesting protein because, so first of all, it has this RNA binding domain here, but it also has this interesting prion-like or low complexity or intrinsically disordered domain. So this is actually a domain that doesn't really fold up into a three-dimensional structure. It's actually quite floppy. And this is because of this unusual sequence that this protein has. And there are many, many proteins like that. But actually, for a long time, we didn't really know what these sequences are doing. And so this is actually, so we focused on this protein. And one of the first things that we did is we just put GFP on the protein. And we observed it in cells. And so you see here actually these structures that in which FUS resides. And this is the raw data. This is actually rendered here, three-dimensional rendering. And you can see that when two of these structures touch each other, that they fuse and into this larger sphere. Yeah. And that was actually surprising for us to see, because I mean, this is very dynamic, this structure. And this fusion actually indicates that we're dealing with something liquid-like here, right? But at that time, everyone thought, I mean, this is also the name gives this away, like granules. Granules, of course, have solid-like properties, and this is what everyone believed. But we saw actually that these structures are actually quite dynamic. And then we wondered, so is this ability to form these kinds of dynamic structures, is that actually encoded in the sequence of the protein? Yeah. And for this, we purified this protein from insect cells, and then we looked at it in a physiological buffer at physiological concentration, and we saw that the protein actually phase separated into these dense liquid droplets here, and they also were very dynamic. They also showed this fusion behavior, and so that was really remarkable to see, because it showed us that the information for doing this is encoded in the protein. And to cut a long story short, now we and many others have published that so these RMP granules are now thought to form by this process of condensation or phase separation. And for this to occur, you need two components. You need typically RNA, but also these RNA binding proteins such as Fas. And they typically have these RNA binding domains, but also these intrinsically disordered regions. And initially, these components are mixed, but then there can be a change in conditions, right? Again, post-translational modification, for example. And then they demix into these dense liquid droplets here. And this is thought to be to come about because of protein-protein interactions, also protein-RNA interactions. And we call these structures now condensates, yeah, because they form through the process of condensation. And so this is now thought to be the principle of how RMP granules in cells form. Okay, now, I mean, this was the work that we did around 2015 to 2018 or so. And but there was this question, of course. I mean, so RMP granules are actually very complex structures, right? And everything that we had done so far in vitro with our biochemistry was just using a single protein. Sometimes we used an RNA, but it was not a very complex reconstitution system. So then, of course, the question arose, can we now actually, given that we know this principle, this physical principle, can we now go in and reconstitute entire RMP granules from the bottom up, right? And of course, entire, I mean, you know, they contain dozens of different components, different proteins, also different RNAs. And can we actually do this in the test tube? And so we focused on these stress granules. And the reason why we did that is that stress granules are very easy to induce. Yeah. So what you see here, this black thing in the middle, this is actually the nucleus. And we have a protein here, which is GFP labeled. This is one of these stress granule proteins. And the cell was exposed to a toxic compound, arsenide actually in this case, at time point 0. I'm just going to run the movie now. So you're going to see that this protein then assembles into these stress granules, and you see this dynamic behavior, these fusion events. So we really wanted to now go about and reconstitute these stress granules. Now, what was known at the time when we started this work? So what was known is that stress granules form when polysomes, when you stress cells, and then what polysomes do is they disassemble, and so they release the large and the small ribosomal subunit, and the RNA that is released from the polysomes is then assembling with RNA binding proteins, also some enzymes like helicases and signaling molecules, into these stress granules. And they have been implicated in translation regulation, also regulation of signaling. So this is what we knew. And that was our starting point. And to reconstitute now these structures, we had to, of course, focus on the key proteins there. And we had genetic evidence that these two proteins, so G3BP1 and 2, are absolutely essential here. Yeah. So I'm just going to show you the data here. So this is the wild type, and this is again cells stressed with arsenide. And you see these structures forming here. But when you knock out these two homologous proteins, there's no stress granules forming anymore at all. Yeah. And so that was actually very important to see, because I mean, there are many other stress granule components, and you can also knock them out, but stress granules would still be there, right? So they may be a little bit smaller, the morphology may be a little bit different, but they would still be there. These were the only two proteins, when you knock them out, they're completely gone. And this was also suggesting to us, okay, these are probably involved in the initiation of these structures, in the actual nucleation of these structures. And so what are these two proteins? So they're homologous proteins. They're quite similar in sequence. So there's a dimerization domain here. It's an acidic stretch, so highly enriched in glutamates. This is actually intrinsically disordered. This is also disordered. Then there's a folded RNA recognition motif and another disordered C terminus here, which also binds RNA. Now, given that we are biochemists, of course, the next thing that we wanted to do is we wanted to purify this protein, which we did here. And to our surprise, we found that this protein, in the absence of RNA, this is a GP-tagged G3BP, is diffuse, so it doesn't phase separate. But when we mix an RNA, then it forms these liquid droplets. Yeah. So this was different to this Fas protein that I just showed you. The Fas protein can phase separate on its own in the absence of RNA. This protein cannot do it. It needs RNA to do so. So there's heterotypic interactions between the RNA and the protein, and then you get these liquid droplets. And then we, of course, wanted to see, so which type of RNA does G3BP actually assemble with? And we found that it prefers long, single-stranded, and also unfolded RNA. Yeah, so double-stranded RNA doesn't work, or highly, secondary structured RNA would also not work. And that was actually very interesting to see for us, because this is exactly the type of RNA that is released in cells that are stressed from the polysomes, right? Because when you read mRNA, what you're doing, what the ribosomes are doing, is you're first of all, you're stripping all the proteins off the mRNA, and you also have to unfold the RNA, yeah? And so these RNAs that are then released during stress, they are exactly this type of RNA. They're long, single-stranded, and unfolded, and this protein had a preference for that. Yeah, so, and this protein is also interesting. I mean, as I said, it's mostly intrinsically disordered. These are the only folded parts. So this NTF dimerization domain, this is the RNA recognition motif. And so we wanted to then understand how this protein actually works. And we teamed up with Rohit Papu here, who did simulations. And in these simulations, what he saw very often is that this acidic domain here and this arginine glycine rich domain were very often close in the simulation. Yeah? So suggesting that there's some kind of interaction here between these two domains. And that also makes sense, because here you have a lot of negative charges, and here you have a lot of positive charges because of arginine. And so you have electrostatic interactions between these amino acids. And so we tested that doing genetic experiments and so on. I'm just going to show you the gist. So what we found out is that this protein can be in two states. Initially, it's in a closed configuration. And this comes about because the positively charged RGD domain interacts with the negatively charged acidic domain. And in that case, the molecule is closed. And it's also inactive because it can't bind RNA when it's in that configuration, or it doesn't bind RNA. But then when you have a local increase of RNA because of stress, what happens is that the RNA can actually outcompete this inhibitory interaction here. Yeah, so the RNA can now bind to the RGG domain, and this opens up the molecule. So it undergoes a huge conformational change. And now it's grabbing the RNA, and then of course now you're assembling the network that you need to form in order to get these condensates. So we have a closed and an open configuration, and this open configuration is induced through the local increase in mRNA concentration. Now, and we think this is now the initiation, so how stress granule assembly is initiated. But then, of course, we know that there are many other components in stress granules that have genetically been shown to be there. And we now wanted to see whether they also can assemble into stress granules. And so we see once you have these G3BP RNA granules, that other RNA binding proteins partition in there, and actually RNAs do so as well. And you see this is specific because GAP doesn't go, yeah, but these proteins all go in there. And so this suggests is that there are two steps. You have these initial liquid condensates that assemble from G3BP and RNA, and then other RNA binding proteins can come in, and then you end up with the mature stress granule. All right, so I think now we can reconstitute these structures really from the bottom up in various complexities. But I mean, there's many other functions you can ask now. And I think one key question in the field is like, what is this good for? Yeah, what's the function of this? And stress granules have been implicated in translation regulation, but it's always been correlative, right? I mean, people have just seen when you see stress granules, the cells also downregulate translation at the same time. But really, it's not been shown that it's causally, involved. And so for this, we actually use an assay here where we, or actually a reporter mRNA. This is nanoluciferase, and we have a 5' UTR, a 3' UTR. And so what we can do is now we can program a translation extract with this reporter mRNA. And of course, it's now translated in our assay. And so we can read this out by reading the luminescence. This is the translation output. This is the luminescence from the reporter. And that gives you a measure of how much translation is going on. And now we now compare two conditions, and you can do this in two ways. I mean, you can do this with salt concentrations. You can also do it genetically because we have a variant of G3BP that is no longer able to form condensates, but it still binds RNA. And this is actually what we used here. And so we're comparing the condition where we have no condensates to condensates, and we're then measuring the translational output. And what you can very nicely see here is that once you have condensates, the translation of the mRNAs is repressed. Yeah, and I think this is now a very good evidence that actually these condensates are really repressing the RNA. All right, so we can now build stress granules in the test tube. We can also look at them functionally. But, and this is an important point, stress granules actually have been implicated in disease, yeah, especially age-related diseases. One of them is actually amyotrophic lateral sclerosis. It's a neurodegenerative disease. And there's one protein, another RNA binding protein, TDP43, which is usually nuclear. But in an ALS patient, this protein forms aggregates in the cytosol. Yeah. And so this has been known for some time. What's also interesting about TDP43 is that it can also assemble into stress granules. So it resides also in stress granules. This was reported here in 2012. And in the field, there's been this idea for a very long time that stress granules could potentially be a precursor for these disease-causing aggregates because these are clearly correlated with disease. Yeah. And so this is actually the question that we now wanted to investigate here, because now we have the ability to form stress granules in the test tube, right? And now we can actually study, is this true or not, this hypothesis? And so yeah, what is this TDP43 protein? So it's another RNA binding protein. Again, it has a lot of intrinsic disorder here. This is also where most of the mutations map that cause ALS. Here are RNA binding domains, RNA recognition motifs. And it's also actually, it has a domain for oligomerization. It's similar to G3BP in that sense. But now what we first had to do is, of course, we had to reconstitute stress granules that contain TDP43. Yeah. So what we did is we mixed RNA, G3BP, and TDP43 to form minimal stress granules that contain TDP43. And so this we achieved here. Yeah, you see that they nicely overlap. And we did then a few tests to see whether our stress granules are really behaving as expected. And so here we deleted the RNA recognition domains, the domains that bind RNA, and then TDP43 is no longer recruited into these stress granules. And the reason why we did that is because this is also what you see in cells. Yeah. So this is TDP43 and G3BP, they reside both in stress granules. But now when you remove the RNA recognition motifs, there's no TDP43 going anymore into stress granules. We did that, of course, to make sure, okay, are we actually reconstituting the real thing here, right? And is the behavior in vivo and in vitro the same? But that seemed to be the case. And then we made a very interesting observation. Maybe I should go back. And so then we looked at this over time. We took our reconstituted stress granules and we looked at the TDP-43 channel and the G3BP channel over time. And we made a very interesting observation. So initially, they're well mixed, both of them. But then something happens here. On the surface, TDP-43 is forming its own phase. You see that G3BP is also excluded now from this phase. And this phase grows. And so what this told us is that TDP-43 is actually unstable in these reconstituted stress gradients. Somehow it initially is mixed in one phase, but then it separates out into its own phase. And we call this actually intracondensate demixing. And this is just a nice rendered representation of this, where you see TDP-43 on the surface here, and this is the G3BP droplet. Now, we then, of course, wanted what is this TDP-43 phase? We did a photo bleaching experiment. And we saw that the mixed TDP-43 is very dynamic. It recovers very quickly, whereas the demixed TDP-43 is barely dynamic anymore. And that was already suggesting, okay, we're dealing with a different state of matter here. It's no longer liquid, it's something else, probably a solid. And of course, given that TDP-43 is known to form aggregates in patients and ALS patients, we suspected that the protein is now misfolded and is forming an aggregate. And to look at that, we actually used a chaperone. This is a small heat shock protein, HSPB1. And this chaperone, of course, recognizes misfolded proteins. And you can see in our reconstitution system that these demixed phases of TDP-43 are very nicely recognized by this chaperone, suggesting that indeed we're dealing here with misfolded and aggregated TDP-43 protein. Okay, now you can say, well, that's all in vitro. Yeah, show me that this is relevant in cells. So what you're seeing here is actually a stress granule, this one here. We're really zooming in here, high resolution imaging. You're seeing the two channels. You see TDP-43 and you see G3BP as a marker for stress gradients. And at time point 0, again, we had a stress here. And I'm just going to run the movie now. And what you will see is absolutely amazing. You can see that even in cells also, you see exactly the same phenomenon. You see that the TDP-43 assembles on the surface into its own phase and separates from G3BP. Yeah, that's really remarkable because it's exactly what we see in our minimal reconstitution system. We also see in this complex in vivo system. And so this, of course, gave us real confidence here that what we do biochemically here is the real thing. Now, I mean, this was a very long story. I just want to show you a few more things here. So now, of course, we wanted to understand what is really going on here. Why is this protein aggregating inside the stress granules? And so we teamed up here with Jitane Mittal, and he's a computational biologist, and he did simulations of TDP-43. Actually, in the condensed state, he can actually do this now. He has incredible computational power. And what he saw in his simulations is that the RM domain, RM1, is actually quite unstable, and it tends to unfold. And it opens up. You can see this here. This is the folded RM domain. This is the unfolded RM domain. And then when it unfolds, there's two cysteines, 173 and 175, which are normally buried inside the RM domain and now exposed to the outside. And you can see this also here, that in this area here, especially when it's condensed, the protein isn't stable anymore. And so this was suggesting that that the protein may be oxidized, that you actually get disulfide bonds because of these exposed cysteines. And so that was our prediction. And especially this also makes sense because the stress granules are formed because of arsenide exposure. And arsenide is actually an oxidative agent, right? It causes oxidative stress. Now, and then we wanted to test this in our reconstitution system. So here's what you saw before, right? So this is the intracondensate demixing of TDP-43. But now we had TSAP, which of course blocks disulfide bond formation. It's a very strong agent that blocks disulfide bond formation, and you do not see the demixing anymore. So it clearly shows that it's the RM1 domain that opens up inside these stress granules and then it unfolds in an oxidation-dependent manner. Now, then, very last thing we wanted to do is, now given that we now had identified these residues as being key, we wanted to now, of course, do genetic experiments and go back into cells. And so this is what you see over time, the two signals, they actually demix. But now when we mutate these residues here, this C173 or 175, you do not see the demixing anymore. So really showing you that we have identified the key regions here that promote the aggregation of TDP43. Yeah, now to summarize this first part of my talk. So we think that RMP granules indeed are condensates, and that the assembly of RMP granules is driven by heterotypic interactions and conformational changes of these proteins such as G3BP1. That TDP43 can be concentrated in stress granules, and that this concentration is actually a problem because upon oxidative stress, the protein tends to aggregate. Yeah, and I think the most important thing for me is that we can now go in and reconstitute disease emergence inside condensates from the bottom up from minimal components. And I think that's really a wonderful thing, because we haven't been able to do this yet. And there's much more we can actually do in the future here, I think. Okay, so this part was about RMP granules. And I have a second part where I want to focus on another type of compartment. That is something that we just published recently. And it's actually also a fascinating story, I think, because in this case here, we, for the first time, reconstituted DNA damage sites. So what you see here is our friend FUS. You heard about this protein before. This is this RNA binding protein. And it's actually a nuclear RNA binding protein. You see 3 nuclei here. And what we're going to do is now we're going to cut with a laser into this nucleus here, and inducing DNA double-strand breaks. And then FUS is going to be recruited to these double-strand break sites. You can see this happening. Now, this was the cut. Here's the time. And you can see now the recruitment of FUS to these DNA damage sites. So this is already what we did back then in 2015. And we had also then, we looked, of course, at the recruitment of FUS over time. This is time. You can see that it's very rapidly recruited. It also leaves with a certain kinetics here within 2 or 3 minutes. And then there's this poly-ADP ribose polymerase. This is actually, this is known from genetic experiments, is another important player here. It's an enzyme which actually comes a little bit earlier than FUS. Yeah, we had looked at that already in 2015. You see that it's already taking off with a little bit faster kinetics than FUS. And so this enzyme arrives at DNA damage sites almost instantaneously. And now what is this enzyme, and what did we know about DNA damage site formation when we started this project? So you have irradiation, you get a double-strand break, and then somehow poly-ADP ribose polymerase is recruited, and it was known that it can bind to DNA or to DNA ends. And then actually what happens is, because it's an enzyme, when it's active, it puts these so-called poly-ADP ribose or PAR chains on itself, but also on other, other proteins such as histones. So poly-ADP ribose or PAR is it's chemically very similar to RNA. But it doesn't really have a sequence. It's uniform in its chemistry. The other interesting thing is that these poly-ADP ribose chains can actually be branched. Yeah, they're not linear, they're actually branched. And so that was known, and then it was also known that there are these proteins such as FUS, which can now bind to these PAR chains, because FUS not only has an affinity for RNA, it can also bind to PAR. Actually binds to PAR with much higher affinity. It's 10 times higher than the affinity for RNA. Yeah, and so that was the state we were at when we began this this study. But of course, there were many questions still, right? I mean, so of course, PARP is somehow recruited here, yeah. But how do you actually prevent that these two ends are now separated from one another? I mean, you have a break, and you somehow must hold these ends together, right? And so how does the cell actually achieve that these two ends are now separated from another and not separated from one another? And so that was actually, for example, one of these open questions that the field hasn't really yet addressed. The other thing that is actually quite interesting about DNA damage site assembly, this is a paper here from Stefan Grill and Stoner Stoyanov and in 2018. It's a very nice paper because what they did here is they looked at all these factors that are recruited to DNA damage. So they did these laser cutting experiments. And they looked at the kinetics of recruitment. Yeah, this is the half-time of recruitment, and this is the half-time of removal. And what you see is that there's groups of proteins coming in over time. Yeah, so you see you have this initial group, and PARP1 is one of them here, which comes in here. And then you have a second group. Here's our candidate FUS. And there's another group of proteins coming in, another group of proteins, and they, of course, and this has been known, but of course, how all of this is regulated, right? No one has any idea, yeah, because we haven't yet been able to reconstitute DNA damage sites. No one's ever achieved that. But I think if we could actually go all the way and then really study how repair is done in the test tube, and this is actually what we're trying to do now. Now, so the key questions here are, so you have a DNA double-strand break, and you have to, of course, first what you have to do is you have to detect the damage. Then you have to find the ends, and you have to somehow synapse them. You have to keep them together. You have to prevent that they separate from one another. And of course, eventually you have to repair the damage, right? And these are the tasks that these damage sites have to do. And so we initially focused on this PARP enzyme, and this is actually quite an interesting protein. It's 1,000 amino acids. It has a lot of domains. There's actually, interestingly, no intrinsic disorder at all in this protein. So it's fully folded. Just has these very short linkers here, but I mean, it's basically a folded protein. You can also see this here. And so you have these zinc fingers, and this is actually the catalytic domain here. And there's these two interesting domains here in the middle. I come to them later. And so we focused on PARP. And my postdoc, Nagaraja, he made this very interesting observation initially. So he just took purified PARP, and he mixed it together with different types of nucleic acids. Single-stranded RNA, double-stranded RNA, single-stranded DNA, and double-stranded DNA. Yeah, double-stranded DNA, of course, has also the ends that you normally have, also like when you have a break. And to our surprise, there was a very specific condensation behavior just only with double-stranded DNA. Yeah, and that was actually quite remarkable to see. And then, of course, we wanted to understand what's going on there. Why does this protein form condensates specifically with double-stranded DNA? And we wanted to really get at the bottom of the molecular basis of this. And this is really, it took us like many, many years to solve this puzzle and a lot of biochemistry. And I'm just going to cut this short here. But we did many experiments. One of them is shown here, which was actually quite disappointing because we made a whole bunch of deletion mutants to see what is necessary for condensation. Yeah, and as you can see, you need everything. The only thing that you don't need is the catalytic domain. Yeah, so that was, of course, didn't help us so much. But of course, some of this made sense because here's, for example, we knew that DNA binding is happening here. Zinc finger 1 and 2, they actually recognized the two strands of the ends. But then we did another really cool experiment. So what we did is now we chopped the protein into two pieces. Yeah, so this is the N-terminal half of PARP with the zinc fingers. And this is the C-terminal half with the catalytic domain and the BRCT and WGR domain. And we mixed it together with double-stranded DNA. And remarkably, when we mix these two fragments, we get condensates. What this, of course, tells you is that none of these fragments alone is able to form condensates. But when you mix them together, there's trans interactions between these domains that you need in order to form condensates. And so that was really cool. Now, we did many more biochemical experiments. I spare you the details. But what we came up with in the end is a very complex or complicated model in the end. So What you see here is how we think about what's happening. So this is one DNA, and there's another DNA, 45 base pair. This is actually what we use in these assays quite often. And this one of these here is a PARP molecule. There's another PARP molecule here. There's another PARP molecule here, and there's another one here. So you see 4 PARP molecules. And we know a little bit the structure. It hasn't been completely solved, but we have some idea of the structure. And so this is what you're seeing here. Now, what we saw in our data is that, so as soon as PARP binds to the end, it immediately dimerizes here. And there's a dimer interface. And once you have the ends connected via this dimer, additional PARP molecules can then somehow, through a lateral interface, communicate conformationally with the dimer. And there's additional interactions here that actually come from these PARP molecules. So this we worked out biochemically. And so then, of course, this then allowed us to propose this model. So we have PARP and we have DNA. And initially, in the recognition, DNA damage recognition process, you have the formation of a dimer. Yeah. And this dimer, there's some interfaces here between for example, the WGR domain and the zinc finger 1 that is required for that. And this dimer mediates the synapses of these two ends. And then we have more molecules coming in that attach laterally to the dimer. And actually what happens is that these molecules can grow along the DNA. Yeah, so basically they can polymerize left and right along the DNA. So we have multimerization along the DNA. Now, and so then, of course, we went to the literature, we did a lot of guessing, but we actually were luckily able to identify mutations that disrupt the dimer interface and mutations that disrupt the lateral interface. And what's really cool is that when we disrupt either the dimer or the lateral interface, we do not get condensates anymore. So you do need dimerization as well as this multimerization along the DNA in order to get these condensates. All right, so everything I've shown you so far was actually these bulk condensate assays, right? These are huge micron-sized droplets. But of course, all of this happens on a single DNA molecule. I mean, of course, in these bulk droplets, we have thousands of DNA molecules. That's not really resembling what's happening in a cell. So we were then wondering if we now have just a single DNA molecule, do we see the same thing? And so here you see, this is a glass slide that's pegylated, and we have actually fixed on the surface here a DNA molecule, and one end is actually unattached. And so one of the first experiments that we did is, so what you're seeing here is now this DNA, and this is the anchor point. This is where it's anchored, and we have some flow coming from the bottom. This is why it's stretched out. And you only see the DNA. You don't see PARP here. Yeah, you only see one channel. But what we did is we at time point 0 we flow in PARP. Yeah, and then what we saw was really intriguing. So the DNA is rolled up by the PARP molecules. Yeah, and that makes sense because you have multimerization of PARP on the DNA, and you have additional interactions, of course, and that rolls up the DNA, and now you have this single DNA condensate down there. Yeah, and so then we put forward this model that, so what's probably happening here is that, I mean, this is the initial situation before the damage. And that's actually important to mention. So PARP is the most abundant molecule or protein in the nucleus after histones. Yeah, it's super abundant. Yeah, and that means that it's probably already scanning along the DNA all the time at high concentration. But as soon as there's damage now, it forms a dimer, sealing the damage, preventing the separation of the ends. And then you also get, of course, because of the multimerization along the DNA, you get a condensate in the end. But we wanted to test this idea. I mean, so far we hadn't really shown that this is really happening also on the single DNA level. So what we did then is, so again, we use our single molecule assay, but now we have both ends fixed on the surface of the slide, of the glass slide. But importantly, we have a restriction enzyme cut site in the middle of the DNA. And so this is our DNA on the surface with one anchor point. Here's the other anchor point. And initially, so what we did is, of course, now we added the restriction enzyme to cut the DNA. And then what happens is, of course, the DNA is separated, right? Because it's cut, and it's just this is where the two anchor points are. But what was really absolutely unbelievable is that when you now do this experiment and you flow in PARP and the restriction enzyme at the same time, you get this. Yeah, so there's no separation of the DNA. And where the cut site is, you get a condensate. And of course, you can always say, oh, yeah, but how do you know it's really cut, right? And of course, we tested this. I mean, you can, when you add a little bit of urea and guanidine to denature the protein, we did this here, that actually DNA comes apart. Yeah. So this shows you that indeed, PARP is able, through dimerization and multimerization, condensate formation, to hold these ends together. Yeah. And we did further experiments in optical tweezer, actually. So what's really interesting is that these condensates even generate forces. Yeah. And so we think that the condensation is important to resist the pulling forces of these two chromosomal fragments. Of course, they're going to pull on this and they want to separate it, but the condensate is so stable that it can now prevent the separation of these two ends. And then, of course, we wanted to see, is that also true in cells? So what you're looking at is here PARP GAP tagged, and this is our dimer interface mutant. This is a mutant where we changed the lateral interface, and you can see that in both of these cases, essentially you have almost no formation or almost no condensation of PARP anymore on the DNA damage side. All right, so that was all PARP so far. But of course, I started this with talking about FUS, yeah? And so, of course, and what I haven't yet talked about at all is what actually happens when this enzyme becomes active, because everything you've seen so far is the enzyme wasn't active yet, yeah? And so just to remind you, so PARP, what PARP does is it takes NAD and it makes these branched poly-ADP ribose RNA-like molecules. Yeah, so this is what the enzyme does, and you can follow this in a very simple assay. So you just run a gel. This is unmodified PARP, and this is when it's modified. You see the smear. This is PARylated PARP, and this is of course as a function of NAD. So increasing NAD, you get longer chains, and you need DNA for that. Without DNA, you won't be getting any PARylation. So you can nicely follow this. And then the thing was, okay, now we have these condensates, and can the enzyme get active inside the condensates? So for this, we used NAD plus fluorescein, which you can actually see. And so what we saw is that, yes, indeed, we got an increase of fluorescein signal inside the condensates over time, suggesting that, yes, they're actually active. The condensates can become active. We can actually block this with an inhibitor. This is important because, I mean, you can always argue, of course, that the fluorescein is just accumulating in there, but we actually do have polymerization inside the condensate happening. So these condensates are active condensates now. But then we made first quite a disturbing observation. We first couldn't make sense of this so much. But so what we saw is that, so this is PARP, this is DNA. You get condensates in the absence of NAD. But when you add the NAD substrate at high concentration of NAD, the condensates went away, they dissolved. Yeah. And we also quantified this. This is here quantified. So you see there's a concentration dependent dissolution of the condensates. And that was a little bit shocking for us to see. Because, you know, first of all, you hold the ends together, forming a condensate, and then you dissolve it immediately. What's the point of this, right? So that raised the question, of course, can these condensates somehow be stabilized? And now I want to bring back FUS, because FUS, we did this already in 2015. when you actually inhibit PARP with an inhibitor, FUS is no longer recruited to DNA damage sites. So it's completely dependent on PARylation. And so then we went back and now wanted to see, okay, now let's bring in FUS and see whether it can actually stabilize the condensates. So this is PARP condensates, PARP DNA condensates in the absence of NAD, in the absence of FUS. Now, when we add NAD, as I showed you before, the condensates dissolve. Yeah, they're not stable anymore. But then when we have FUS around, they are stable. Yeah, and you may ask, why do they actually dissolve? We also understand this because what's happening is that when the enzyme becomes active, it PARylates itself with part chains, and these part chains, they look a little bit like DNA, and they can compete with DNA binding to the zinc fingers, and that's why PARP lets go of the DNA. But now we have a FUS, and FUS, of course, is able to bind to PAR. It can cross-link PARP molecules by binding to PAR, and it can actually stabilize these condensates. Now we have catalytically active condensates that are stable, and well, we still had to show, okay, again, is that also happening in our single molecule assay? Now this is without NAD. I showed you this before. You have these PARP condensates. This is DNA, and this is PARP. And there's no, I mean, actually, you can see that in the absence of NAD, FUS is not recruited to the condensates. But now when we add NAD and the condensates become active, FUS is recruited. And also there is no separation of the ends. Yeah, because I mean, we know it's cut, but they don't come apart. So FUS is somehow able to stabilize these condensates. So now we have this model for DNA damage site assembly. So you form this initial condensate, which is almost like a glue, right? It holds these ends together. But then, of course, what you have to do is you have to, because it's so gluey, it's so solid, like you have to make this dynamic compartment, because if you want to bring in now the DNA repair enzymes, you have to have a dynamic compartment. And this is the job of the enzyme activity of PARP. So it PARylates itself, and then this brings in FUS. And actually, we've done photobleaching experiments. This structure here is much more dynamic than this structure. You photobleach this, there's almost no recovery. You photobleach this here, it's rapid recovery. And also here, this is indicated here. So the DNA ends are now free because PARP lets go of the DNA. And this also means now that the repair enzymes can now come in and repair the damage. And that's what we're working on right now. Okay, so to summarize this part, so PARP can form co-condensates with DNA through DNA end binding and then multimerizing along the DNA. And these condensates, they really glue DNA ends together to ensure the synapses of the ends and ensure also mechanical stability. But then PARP self-PARylates inside the condensates, and first what it does, it promotes the DNA release, but also recruits FUS, which then stabilizes the condensates and also fluidizes them. I mean, I didn't show you the data, but as I said, this becomes much more dynamic there. And these condensates are now primed for repair, right? And so, and this is, again, the most important thing for me is that we can now go in and really reconstitute from the bottom up active functional membranous compartments. And yeah, and I think this is really, again, very nice, and especially for the DNA damage field, this is going to be very nice to develop this assay or these assays further to better understand mechanistically now how DNA damage works, because actually we have a lot of genetic information on DNA damage. We have inhibitors that we can use to block certain molecules there, but what's really happening biochemically, the step-by-step handover, yeah, is really not understood. And I think this is another way to understand this with this system. Yeah, and with this, I'd like to thank a lot of people. Everyone here in yellow has contributed funding sources, and I'm happy to take questions now. Yeah.
Yeah, thank you. Very beautiful work. I have a question about your last experiment.
Yeah.
You said that PARP1 doesn't need the catalytic domain to form condensates, right?
It doesn't need what? Sorry.
You don't need the catalytic domain of PARP1 to
To
form
forming
the
the condensates. Yeah, exactly.
Did you do the last experiment where it forms the condensate in the double strand break using that mutant without the catalytic domain? See what would happen?
Yeah, so in the last experiment you're referring to
Is
this
that
here?
one? Yeah.
Yeah, so that actually we haven't done. But I mean, if you we could, well, we did this in the tweezer. So we did tweezer experiments where we actually removed the catalytic domain. And so that still forms condensates, yeah. But of course they would not be able to then parallate themselves, right? And then you wouldn't be able to go further. And so basically you're stuck in the glue state, and you wouldn't be able to proceed to the next step, which is the more dynamic state. Yeah. And this is actually, I mean, something that some of you may actually know this. I mean, so PARP is it's actually involved in cancer. Yeah, and so there's now FDA approved drugs, 4 FDA approved drugs that target the enzymatic activity of PARP. And so what's really interesting is that what these inhibitors do is they stabilize this glue state. Yeah, so this is something we're working on right now. And so you have this glue thing sitting on the DNA, and this is sort of like a roadblock now for replication. So the cancer cells can't go through this anymore, and that's why the cancer cells die. Yeah, because they cannot get out of the glue state. So you need the enzymatic activity in order to get out of the glue state. And of course, if you get rid of the domain, yeah.
And just another quick question of ignorance. So you said that to form condensates, all the proteins need a DNA or RNA binding domain. So all the proteins that
Yeah,
don't bind any nucleotides.
that's a good question you're asking, and I think it's a very important one, because that's exactly what we've seen. You know, there's very few condensates that are protein only, right? I mean, there's some T-cell receptor signaling condensates, maybe they may be protein only, but pretty much all the other condensates that we looked at so far, there's always a polyanionic nucleic acid involved, right? It's either DNA or RNA or PAR. It's always one of the three, yeah, which is the key scaffold molecule on which these proteins assemble. And so that's something that the field is only realizing now, right? Initially, the field was very protein centric, but I think that's something that we have to forget about. That this is really the nucleic acids play a fundamentally important role here, and because they're long molecules with many binding sites, and yeah.
Could it be possibly outside the cell, say in a plant cell, cellulose or any other polysaccharides of the cell?
Yeah, so of course, anything that is this, usually multivalent could of course take the job. Yeah, and yeah.
Thank you.
Hi, and thank you for the seminar. I have I have 2 questions. The first one is, what about the environment inside the condensate? I mean, do you know what components or factor can alter for better or for worse the DNA repair mechanism machine? I don't know.
So which factors could alter it, or
In the environment inside
in the environment, you mean? I mean, so whether there's environmental effects on the DNA damage sites, you mean? Yeah, chromatin, for example. That okay? That's obviously chromatin. Yes, I mean, but that's something. I mean, because I mean, this is something we didn't study here, right? That normally DNA is wrapped into histones, right? And so that's, but yeah, and so that may be an important factor here that we still have to study. But there are many other components that we haven't looked at yet, right? Yeah.
Okay, thank you. And the second one, there's a thing that I don't really understand. How do the DNA fragments get arranged inside the condensate?
Inside the condensate?
To maintain the correct assembly order. I don't know if you need like some type of factors that order these small pieces, or they, I don't know, follow some.
I mean, you have to understand that the small pieces is a little bit artificial what we're doing here, right? I mean, this is these 45 base pairs, or we also use the 100 base pairs. Of course, normally you have just a long DNA molecule, right? And so that's actually the real deal. But I mean, how this is organized inside these huge condensates, I mean, we can only guess. But I mean, I showed you this one slide, you know, maybe I should go to that. And this is at least how we imagine it to be. But of course, right now we're doing cryo-EM actually to look at the actual structure, but this is going to take a while to actually really see it. But the idea is, you know, you have these dimerization, you have these molecules sitting here laterally, and then there's interactions here to other PARP molecules that are sitting on other DNA molecules. Yeah, but the important thing is like PARP only interacts with PARP when it's bound to DNA. PARP does not interact with itself in the absence of DNA. Yeah. So, but how this is really organized, we have to wait for the cryo-EM structures. Yeah. It's going to be interesting.
Thank you.
Yeah. Hi. Very nice talk. It's my turn? Yes. Well, I have many questions, but I'm just choosing one. When you refer to stress granules, you refer to arsenide-induced granules or heat granules.
Yeah.
So my question is whether all granules are the same? The first question, and the second question
there's thousands of different types of stress granules. This is the problem. I mean, they all have different compositions, and arsenide also, of course, is not a very natural kind of compound. I mean, I give you that, of course, yeah. But I mean, we tried many others, and any oxidative stress actually induces stress granules. And yeah, you're completely right. I mean, like, there's probably, depending on what stress you have a different type of stress granule, which is different composition. And what we have no way right now to understand how this actually works, right? I mean, no one really knows.
Yeah, I know that. But so then the next question would be whether G3BP1, which seems to be essential for the granules that depends on arsenide addition, is like an essential protein for all stress granules. Or is
all except for the ones formed by osmotic stress.
Okay.
Yeah. So, but all the other ones need. Oh, actually, heat is also not really required. I mean, it depends again.
Okay.
So, but it's absolutely required for, let's say, oxidative stress conditions. Yeah.
So in the case of the oxidative stress that you talk about, you said that the TDP43 depends on the cysteine bindings to form this intracondensate.
Yeah,
no?
Actually, so yeah, I can. There's a longer story. But maybe you could finish your question.
Yeah, and so my question is related to whether TDP43 is rather is also interacting with other proteins that also are getting denatured or aggregated because of the oxidative stress. And actually the reason, because now you see the cysteine bound is because it's not interacting anymore with the other elements there.
Whether it interacts with other proteins, that could be. We have to look at that actually. So we don't know yet. But it's very well possible if other proteins have cysteines, that they could also be affected by that. But maybe I should also tell you, I mean, like, there's much more to the story, which is actually quite interesting. So you may wonder, you know, I mean, why is it aggregating inside the stress granules? Yeah. And what we found is that, first of all, it's concentrated in there. So it's a much higher concentration. But the thing that we realized, and this is, I didn't show you the data, but this is actually quite intriguing. So TDP43 binds specific types of RNA, UG-rich RNA. It likes to bind UG-rich RNA. But this type of RNA you only find in the nucleus because it's actually in introns. And you would never find that in the cytosol. Now, but when the cell is stressed, what happens is that they shut down transcription and they make less introns, like less splicing going on. And so the substrate for TDP-43 is no longer in the nucleus, or it's at lower. And so then it shuttles out into the cytosol. It looks for RNA.
And
Yeah,
it's not.
of course it binds any RNA, but it binds UG-rich RNA much more. And so then what we found is that, so the RNA that you find in stress granules is mostly not UG-rich. And so TDP-43 binds to this RNA with a very low affinity. Yeah, it's a very high off rate when it binds to this stress granule RNA. And so what this means, so now you have TDP-43 highly concentrated there, but it also has a high off rate. It comes off all the time, on, off, on, off, on, off, but it's also highly, and so then, and that's actually why it happens there and why it doesn't happen in the nucleus. And so, but it's very nice that we can now explain why this actually happens also on the molecular level.
Right. Yeah.
But this could also be happening for, I mean, we're actually looking at other RNA binding proteins, whether they also are destabilized inside because it's not the right RNA for them anymore inside. Yeah.
Exactly. So yeah, and actually a more general question is that, of course, most granules or most biocondensates are very complex. No, they have a lot of proteins inside.
Yeah.
And of course, well, maybe RNA or and DNA. So when you describe this, and you said that a protein can form biocondensates in vitro, and then that is, I mean, the characteristics related to that are the ones that are important in vivo.
Yeah.
The question would be whether there are additional things that when we do an in vitro experiment, you do not consider, right?
Yeah, I mean, there's always, of course, things that we could be missing here, right? But one thing that we're always trying to do is, we go back and forth between cells and in vitro, and we see a very nice correspondence actually. And every time we see a correspondence between what's happening, then we're quite convinced that. But I mean, one danger, of course, and I totally agree with that, is that when you're doing biochemistry, all kinds of things can happen, and you just have to be very careful because you may be studying some artificial stuff, and you really always have to go back into the cell and make sure. Also, then I mean, the best way to do this is you use your condensate in vitro system, and you identify mutations that disrupt the condensate in vitro, and then you go into cells. And if you then disrupt the condensate also in cells, I think that's pretty solid. Yeah, it's not easy, but that's the path we should all take. Yeah, in my opinion.
But because we can discuss this, but because valency is involved in this, and the interactions can be between inside the same protein and with other proteins.
Yes, absolutely.
Maybe those mutations.
But that's the thing that we also more and more see. So I mean, when the field started, we all had this idea that it's mostly homotypic interactions, right? FAS interacts with FAS, TDP interacts with TDP, and that's how it's done. And what we now see, it's all heterotypic, right? It's different proteins interacting with different proteins, different types of proteins. That's mostly what we're seeing now. And so the field also we have to change a couple of things that we all believe for a very long time now, for 10 years almost. And but that's also exciting now. I mean, these heterotypic interactions are not well understood at all. But this is actually how it seems to be. Yeah.
Exactly. And the last thing is just there are also condensates, metabolic condensates that don't need that DNA or RNA, right?
Yeah, of course. I mean, metabolic condensates are super interesting. I mean, but there's a lot of people who think that in the metabolon idea, right? So that even glycolysis or so is organized into condensates, and that's something that I know people are actively working on that, but it's not easy.
Right.
Okay, so somehow some of my questions have been already formulated, but anyhow, I'm going to ask. When you perform the condensate in vitro experiments with PARP, so you're using naked DNA. Have you been able to use chromatin instead of naked DNA? Because at the end, in the nucleus, the DNA is in the form of chromatin. It's forming chromatin, and one of the signals is known after double strand breaks is the phosphorylation of histone H2X.
Absolutely.
Right? So then I think it will be very interesting to see what happens if you perform the same experiments with
so we're doing this now. We have a collaborator and he's actually able to form chromatin in vitro. And so we're actually doing these experiments. I can't tell you yet how it's going, but yeah, that's the next question, right? What and also because you can correlate also histones, and you probably also have to remove some histones there. Yeah, so absolutely key question. And the other thing is actually, I mean, so many things. The condensate could also be involved actually in somehow removing the histones, right? Mechanically. I mean, maybe that's also a possibility, right? Actually, one other thing that we also think the condensates could do, which is also very interesting, is so what happens as one of the first steps during a repair is that there's a resection. So you basically remove one strand and on one side and one strand on the other. But it's never been known how far. Why does the enzyme stop somewhere, right? And our idea is, for example, that the enzyme is only active inside the condensate, and this is also how the cell limits the repair to this portion of the DNA, and it doesn't go further anymore, right? You limit it to a certain area of the DNA, and yeah, all of this is important. But
okay, so the second question is quite simple. What's the physiological response of the cell if, for instance, you knock out RBP, G3BP, for instance? What happened with the cells? If you put the cells under stress, but you knock out the gene for G3BP.
Yeah.
What happens
so
to the cells?
what's the defect actually?
Yes. What's the physiological response of the cell?
The problem
is it essential?
Yeah, so the problem with G3BP is that it has so many functions. And the interesting thing is that actually you get also immune defects, because I mean, what I didn't tell you is that G3BP is also involved in other types of immune condensates, like innate immunity. And so when there's nucleic acid sensing going on, like CGAS, RIG-I, all these nucleic acid sensors, they also seem to be working to some degree with G3BP. And that seems to be a big problem. When G3BP is no longer there, they can't form condensates anymore. And then you have immunodeficiencies. So that's one of the phenotypes that you see. But it's also involved in signaling, and I mean, it definitely is also essential, right? When you knock it out, and you can't make a mouse out of it, right? When they're both gone. Yeah.
Thank you.
So we have a question by Dr. Cesar Cuevas, and he asked, so given that TDP-43 aggregation occurs late in life in the patients, perhaps this is due to different changes in the environment of the H neuron. So are you interested in using the in vitro reconstituted stress granules to test the effect of a different environment?
Yeah, so I mean, of course, the environmental that there are environmental changes that, of course, also trigger ALS. This has been around for, and I think oxidative stress is probably the one that is most important here, right? I mean, we actually do believe that it all gets started with oxidative stress, and we know that neurons, especially H neurons, are more and more, I mean, on oxidative stress conditions. But this is only one step, right? So there's actually there's multiple steps that lead to the aggregates in the end, which cause the disease. And we have only reconstituted the first step, which is like we have these amorphous aggregates, but then actually what you later on you get these highly ordered prion-like, amyloid-like aggregates, right? And so this is another thing that we have to see. But we also do see that these things form from these amorphous aggregates. But anyways, I mean, so yeah, environmental effects can be studied also in vitro, but for now we focus it on oxidative stress.
Okay, and his second question is based on the affinity of TDP-43 for GU-rich RNA oligonucleotides. The question is whether RNA GU-rich oligonucleotides can be used for treatment of the neurodegenerative diseases.
Yeah, of course. That is actually, well, a colleague of mine, Jim Shorter, actually, and there's others who pursue this right now. Yeah, and they actually found RNAs that bind very, very strongly to TDP-43, but the problem is they bind so strongly that they never come off. So that you also kill the TDP-43, essentially. But so that's one possibility, I think. And I mean, there's also our data shows that the RM1 is the one where it all starts, the RNA. And so as soon as RNA is bound, the RNA recognition motif is stable, right? So indeed, RNA would be the way, or some RNA-like molecule. Actually, I mean, I'm involved in a company, Dewpoint, and we've found a very interesting compound that actually does exactly that, right? It's not RNA-like, but yeah, that is definitely very interesting.
Okay. Is there any other question?
Hi. Hi. Oh, me first? Oh, okay. Hi. So my question is about how might the microenvironment within the condensates I mean affect the encapsulated molecule?
Yeah, so that's a very good question, the microenvironment, because that's actually where the field is excited about this right now a lot, right? So